Your career begins here at Satyabama Institute of Science and Technology, a deemed to be university, a nationally recognized and globally acclaimed destination to accomplish your academic aspirations. The Colonel Dr. JPR Research Park, along with the International Research Center, houses advanced research facilities that have 13 centers of excellence, 10 plus technology transfers, 100 plus patents, 10,000 plus reputed publications. The Satyabama Center for Advanced Studies is a state-of-the-art facility built with the aim to cater to the futuristic aspirations of our students with the aid of a metaverse studio, drone labs, makers space, media center, advanced characterization facilities, ICT-enabled classrooms and supercomputing laboratories. The engineering departments are equipped with sophisticated infrastructure fulfilling the academic and hands-on requirements of our students. The architecture department is furnished with sophisticated design studios and spacious drawing halls, all of which support the learning process of our postmodern generation architects. The Satyabama General Hospital, along with the School of Dental, Nursing, Pharmacy and Physiotherapy, have advanced clinical facilities. The monumental Central Library houses a diverse collection of books, periodicals and journals. Additionally, it also has an open-air theatre with a seating capacity of 1,000. The Management, Arts, Science, Law and Hotel Management departments comprise of all the necessary and required facilities, creating the perfect ambience for our far-sighted students and catering to their skill set needs with the end goal of equipping them to live their dream. Our chain of laboratories with practical exposure equips our students to build a successful career. Our institution has a team of dynamic and outstanding teachers who are ever reachable to our enthusiastic students, promoting the spaces to learn and places to grow. Greenery around the campus greets and accommodates our students, who are the backbone of a developed nation, edging them on to achieve the summits of their success. Satyabama Institute of Science and Technology,
A very good morning to all. Thank you for joining us today. We are here gathered for the Ocean Research Knowledge Series 2026, a 30-hour lecture series organized by Center for Ocean Research and National Facility for Coastal and Marine Research, delving on the research insights from the Center for Ocean Research and the NFCMR, Satyabama Institute of Science and Technology. I heartily welcome one and all joined here today. Today, we have with us our speaker, Dr. R. Srinivasan, who is the Assistant Professor Research at Center for Ocean Research, and he's here to talk about eukaryotic cells and metabolic engineering. Welcome, sir. Over to you, sir.
Is it visible?
Yes, sir, it is visible,
Okay.
sir. Thank you, sir.
Okay. Thank you. So today we are going to see on the topic of synthetic biology and metabolic engineering approaches in eukaryotic systems. So first we'll be going to the introduction section, like synthetic biology and metabolic engineering role in eukaryotic and prokaryotic systems. Synthetic biology, which is nothing but what the tools we are using, like what are the constructs we will be using for like promoters, terminators, and operon structures and mRNA secondary structures, translation efficiency, translation rate, and mRNA stability for designing the whole construct. The way, the tools which we'll be using for engineering, that is metabolic engineering, to enhance that pathway through by overexpression or deletion or flux balance, to in order to enhance the flux. Even we have to be modeled to it, and from these results will be go for productivity, yield, and growth rate. What is that will be? So coming to the prokaryotic versus eukaryotic, we know that prokaryotics are bacterial systems mostly, and eukaryotic systems are, it's versatile, like yeast, insect, mammalian cells, even plant cells. This and all which is comes in eukaryotic system. We know that so when in case of yield or the fast of the yield, so it will be better in the case of bacterial system like prokaryote, but in the case of eukaryotic system, it's a better in the case of yeast and compared to other systems. Yield wise also, which shows that prokaryotic is a significant increase, but in the case of eukaryotic system, it will be better in the case of compared to prokaryotic system. Even post translation modification also, which will be better in the case of eukaryotic system and compared to prokaryotic system. It's because of it has a like kind of human cell, so any modifications happens after protein translation also, which is easy. For example, like glycosylation, that all will be easier in the case of eukaryotic system. The cost wise, if you see that prokaryotic system is far better than, significant better than that eukaryotic system, because of it is fast growing, and even the media compositions which is required for it is a cost effective when compared to eukaryotic system. So application wise, it will be go for protein eukaryotic system, simple proteins and all we can do it in prokaryotic system. Whereas in the case of, as I said, that post-translation modifications like disulfide bond protein or glycosylated protein, this one all which is comes in post-translation modification proteins. So in the case of similarly, even if you take a yeast expression system, you can go for membrane protein, large size protein, viral vaccines, signal proteins, cytokines, kinase. Similarly, mammalian cells also, which plays a maximum major role in case of post-translation modifications. So in this case, we'll be going to see today that only mainly on eukaryotic system on microalgae. So why we have chosen is microalgae is far better than the other eukaryotic system when compared to the media composition wise, it is more cheaper than compared to all other eukaryotic system. Even you can grow it large scale at the outdoor cultivation when compared to other eukaryotic system. You see that yeast or mammalian cells or plant cells, initially you have to go for controlled closed systems. But in the case of microalgae, it is not the case. So these are the main advantages of microalgae. So microalgae, we know that it is a phytoplankton. It's a small, very small, like single cell plant-like plant organism, which is around 1 to 550 micrometers. So it is altogether, which when it say about larger size, it is the multicellular, it's a seaweed. In the case of smaller one, we say that microalgae. So the major type of microalgae, which is classified into red algae, green algae, brown algae, and diatoms, blue-green algae, and dinoflagellates. So these are the major types of microalgae. So what is the use of this microalgae, which is involved in different fields like biofuel? Biofuel means which it can we can say it as in biodiesel, biobutanol, bioethanol, biogas. These are all biofuel. Same thing, it will be useful for bioplastics also, which is producing the PHA molecules like polyhydro. So this and all, it's a bioplastic which just comes in. It also acts as a feedstock for biofuel production also because it has a high carbohydrate content. So similarly, we can see it in the cosmetic industries also, like pigments, which is plays a major role in the case of cosmetic industries. Similarly, this can be used as an cattle feed or animal feed. We can be an alternative for live feed also for aquaculture side also. So it also has that high nutraceutical values like pharmaceutical values like vitamins and even nutraceutical products like omega-3 fatty acids also. So we know that in foreign countries they are using it as a feed food also. So in India it is like slow process. Similarly, from this, they are using as a fertilizers like carbohydrates. They are utilizing like it also acts as a biostimulants for the growth of the plant. So they are using as a fertilizers. These are the commercial use of microalgae. So when you compare to that, so far the tools available, like synthetic tools available so far, and based on that we have classified these. These are the stains they are using currently on commercial production also, as well as in the case of basic research also. So in that species, mainly on Chlamydomonas renati, Nanochloropsis, Phaecoidoculum, Thylacosura. These are all main tools is available for whatever the genetic modification you want to do, even metabolic engineering to enhance the flux of the metabolic pathway. So that are all these are the tools which is available, easily available. We can target to for the production of byproducts. So in you take the commercially important metabolic pathways in the algae or mainly on we can go for recombinant protein in the chloroplast, nitrogen production through photosynthesis. And if you see that biomass, we can use as a we discussed that feedstock for even for the syngas production, nutrition, and aquaculture side also. Similarly, we can go for this carbohydrate from the algae for ethanol production and lipids and fatty acids for biodiesel productions. And if you see that FUFA content and astaxanthin, it is on a nutraceutical property, carotenoids as well as omega-3 fatty acids content. And if you take it, the precursor molecule isopyrenons, we can use as a gasoline. So we are using with the different methods like organic compounds, like nutrients, these and all, which is plays a major role for the conversion of these products. So, however, there is a limitation and challenges and bottlenecks through in microalgal stain improvements. So, mainly on when you go for screening of large scale
Link to
screens or
URL.
involved in user large manpower.
Thank you.
So there may be a chance of these mutants. If you go for physical development of mutants or chemical mutants, there may be a chance of reversion of these mutants, and it may be cause the interrupt in the case of production wise also. So another major issue is like after constructing these protocols also plays a major role in large scale production. Sometimes upon large scale transfer, the strain may be experience the adverse conditions like temperature, pH, and what the environmental factors which is plays a major role. But when you see that when compared to this controlled conditions which you are providing in the lab conditions, so in such a way that you have to design the photoreactor, it should not affect the cell behavior and fitness. So if you say that, as I said, that we can go for mainly we will be trying with the reverse genetics, which is nothing but towards the targeted proteins we have been modifying and to enhance the strain improvements. So this reverse genetics, we using this synthetic biology only. We are saying it as an a little bit costly, but it will be gives the promising results. So you need appropriate equipments for that genetic transformations also. So in that way, we are going to see today two case studies that which we are going to see on production of this phytoin by inhibition of phytoin desaturase in marine algae, particularly Donella salina, via RNA interference. It's nothing but gene silencing. So we know that carotenoids is a major pigments which is found in the nature, and it is also present in the all photosynthetic organisms. So carotenoids have the key roles in the life as well as in the commercial importance. It has plays a major role in functions in photosynthetic activity in the algae also. It acts as an accessory pigments. It also precursor molecule for pheromone production in the algae, and it also plays a major role in higher plants also, like seed dormancy, embryo maturations, germination, cell divisions, floral growth, and stress response. So it also acts as an antioxidant pigments, so in order to overcome the stress in the algae. So we know that phytoin, this is the pathway for carotenoid production. So we can able to see that at the precursor molecule, which is after geranyl diphosphate, is a phytoin is a major molecule. So it is an precursor molecule for all the carotenoids. So it is a 40 carbon intermediate compound. So it has a 3 conjugated double bonds, and it's a colorless carotenoid, because as this colorless carotenoid will be further converted into through sequential reactions into colored products like lycopene, beta carotene, and xanthophylls. So in order to produce this, we are going to target in this work like PDS, which is nothing but phytoin desaturase gene. So the importance of this phytoin is more stable carotenoids when compared to other carotenoids like beta carotenes. It is commercially important in cosmetic industries because as it is colorless, so mainly they are using for cosmetic industries. So in order to, it has a several advantage, but in case of cosmetic industries, we need to be protect the skin. So in that way, it will be using as an UV observer, antioxidant, and anti-inflammatory agent. So there is a product, still they are working on the product on production of this phytoin by Israel Biotechnology Company. So in this company, they are using it as an, there are different products which is established in 1995. So they are using different source from tomato also they are working on, from the algal also they are working on this for producing this colorless carotenoid. However, this production, which is majorly they are depend upon that herbicide based inhibition. So in order to overcome that herbicide usage, so we are trying to target gene silencing way to production, enhance the production. So for that, we are trying to use RNAi mechanism, which is nothing but we know that once it is RNAi like kind of mechanism, once it's entered into the body, even the algal cell, the dicer complex will come and it will be forms dicer and this complex. It will cut into that mRNA, it will cut into pieces. That complementary strands will be go and sit on over the mRNA to stop the inhibition. This is the overall mechanism of RNAi. So we have here we have constructed with gateway technology. It is nothing but the exchange of fragment from entry vector to destination vector. Entry vector here is just a cloning vector. It is not expression vector. The destination vector is the algal vector for expression of our particular protein or gene constructs. So there will be initially will be cloned into entry vector. Then will be LR recombinase enzyme will be adding to exchange of this fragment into that ATR fragment. So we have here ATTL site in the destination vector ATTR site. So in that way it will be exchange the fragments. In that way we have targeted towards marine microalgae like Donella salina. Particularly it is a high salt tolerant carotenoid producer, highest carotenoid producer. The main advantage is little bit it's a fast grower and compared to the other marine algal strains. It can able to survive from 1 to 11 pH, and the salinity also it can able to survive from 3 to 31%. Temperature also it can able to survive from 0 to 38 degrees Celsius. So in order to gene silence this PDS gene, so we are targeting towards this is the construct what we have here. Like it will be like sense construct and as well as antisense construct will be there. So it will be forms a loop like structure, like hairpin like structure. Then it will once it is entered into the side. So as we seen before the slides that RNA technology, once it is the hairpin loop enter into the cell, so it will be this complex will be formed. So there it will be cut into pieces. So that complementary strands will be go and sit onto that endogenous mRNA sequence. So in that way it will block the protein. Transcription will be stopped. So translation will be obviously it will be get stopped. So first we will be seeing that how this initially we plan for agrobacterium mediated transformations. So at the time while we are working, we don't have the sophisticated equipments like electroporator or biologistics. So in that way, we are targeting towards agrobacterium mediated transformation. So this is the overall protocol for agrobacterium mediated transformation. Like secondary structure will be secondary culture will be culture it, and then will be plate like a solid medium will be plate like a lawn culture. After that, we'll be side by side the harboring that RNAi PDS construct harboring agrobacterium will be co-cultivated with Donella salina. And for the 48 hours. After that, we will be using cefotaxime wash. Here, the step which we'll be using for to remove that agrobacterium alone, it should not affect the algal. So you need to before that you need to check that toxicity effect of cefotaxime also. What your selection markers you are going to use for. So here we have used selection markers as acromycin as a selection marker. So in that ways we will be adding into the selective medium. So for after 6 to 8 weeks, we'll be finding the colonies. That colonies will be separated and will be characterized for further analysis. So these are the 17 colonies we have isolated. So we have isolated the DNA from all the 17 colonies, and we have checked with the DNA integration in the genome. So we found that around 6 to 7 colonies were found that positive. That alone we have took it for southern blot analysis, which is nothing but how many copy numbers of integration of this transgene, whether it is present in the genome. So in that way, we found that all the transgenic line which has a single copy number. So further analysis, which is for RNA expression analysis by northern blot. This is kind of how we are doing for southern blot. Same way we will be here. The probe only will differs against a particular gene. So when you see that transgenic lines, which shows that PSY, it is upstream of the pathway which is present in PSY, which is the geranyl pyrophosphate will be converted to phytoin. For that, this PSY will be involved. So our target is PDS. So when you see that transgenic line is almost 70 to 80% of suppression we have observed when compared to the wild type. Similarly, is lycopene cyclase B, which is downstream of the PDS gene pathway. So that also which is almost suppressed in compared to the wild type. Even cyclase carotene hydroxylase also, which is reduced in the case of transgenic lines. This acting as an endogenous gene. So we have observed that a similar pattern of expression in all the transgenes. So next one will be after seeing the expression pattern and the molecular level characterization, we have seen that carotenoid accumulation in the case of PDS mutant lines as well as in the case of wild type. The C which is mentioned here is the control. The RNA line which shows the arrow marks which shows that different RNA lines that shows the suppression of all the carotenoids in the present in the algae. Similarly, because it has a different wavelength, carotenoids is a 450, but in the case of phytoin, we need to look for wavelength of 296. So it is a colorless as it is. So the wavelength this varies to 296. So for that, at the time we have seen that presence of transgenic lines in the phytoin. Whereas in the case of control, we didn't observe the phytoin because it may be the chance of it is a sequential reaction. So phytoin will be converted into the carotenoid in the control. Whereas in the case of transgenic lines, it shows that phytoin accumulation. So we have calculated quantified the phytoin level and the other carotenoids also. If you see that phytoin level, which is not observed in the case of control, whereas in that highest we have observed in the transgenic line 5, RNAi line 5, around 108 μg per 100 mg of dry weight. So even similarly, we have seen a significant increase in the case of phytoin. Whereas in the carotenoid level, the RNAi lines was decreased is because of down regulation of this PDS gene. So even the lycopene also, which is decreased in the case of RNAi lines, lycopene also decreased, and even lutein also it's a main components of carotenoid, which is also decreased in the case of RNAi lines. This shows that phytoin production we have observed in the RNAi lines when compared to the control. So after that, we have seen that how the phenotype which is response in PDS mutants. So in that way, we have observed that decrease in the growth rate of all the transgenic lines compared to the control. So I will say the reason why, what behind it. Even the chlorophyll level also, which is affected in the RNAi lines. We have seen that sensitivity of high light and low light condition also. We observe that this transgenic lines not able to survive at high light conditions. So even the photosynthetic activity is also decreased in the case of RNAi lines when compared to the control. So the overall it has an maximum reduction of PDS in carotenoid level. This RNAi 5 lines which shows that maximum phytoin content of 108 mg per 100 mg of dry weight. So the phytoin accumulating phenotypes exhibited lower growth rates and high sensitivities to high light conditions. This is mainly because of partial, because we didn't suppress completely. So partial suppression of this PDS gene leads to chlorophyll. As I said that it is an also which is involved in the electron transport, the photosynthetic activity. As I said, it is also access on accessory pigments for photosynthesis. So this also leads to chlorophyll degradation, bleaching effect in the highlight condition. So it also causes malfunction in the photosystem. That's why we have observed that photosynthetic activity also decreased. So especially in phytoin accumulating cells, this reveals that PDS acts as a major control point for carotenoid synthesis as well as for photosynthetic activity. So in order to see that it is an one of the which it is not which is comes in algae, but even though I want to show you that how it is response in the plant wise, we have studied same kind of studies in, but we have used in ZCDS genes. So here is the zeta carotene desaturase gene. So here also same technology we have used, same vector system also we have used. In that also we have found that phytoin accumulation high in the tomato. So in compared to the plant also, which is growth are higher in rate. So even the expression pattern also, which is carotenoid level was decreased in the case of different stages of fruit also we have seen that mature fruit, breaker stage and ripen stage also we have seen it. So even the expression pattern also, which is observed in the PSY and PDS also, which is enhanced, but in the case of PDS and ZCDS and this and all, which is present in the upstream of the pathway, which is decreased here. So in the overall case of RNA lines, which shows that carotenoid level, which is decreased, and the phytoin accumulation we observed in the case of fruit and as well as in the case of leaf also, we observed more amount of phytoin accumulation when compared to the control. Even main flavonoids and chlorogenic acid, which is in the main components of the flavonoids, which is present in the tomato. So that also which is decreased in the case of RNA lines. So even the case of chlorophyll, as we seen in the microalgae, similarly effects we have observed in the case of plant also, like chlorophyll disruptions also we observed, and even in the tomato, the carotenoid accumulation was completely disrupted. As we seen that the plant size also which is increased, so that we have seen here, like it is due to that gibberellic acid which plays a major role in the shoot development. That's why we have observed in the RNA lines which observed that higher growth rate. Abscisic acid was decreased in the case of, because it has an acts as a, so as I said that it is a pheromone which is phytohormones which is a main precursor molecules of carotenoids. So that as it is decreased, the abscisic acid also decreased. This also shows that the CDS plays a major role in the case of carotenoid level in the case of crop plants, particularly in tomato. And next study will be seeing in the algae, similar like here it is the target of lipid production as well as biomass enhancement. This is another study where we have expressed this ACC deaminase gene in Chlamydomonas rhinorti under nitrogen starved condition. So we know that ethylene plays a major role as a gaseous plant pheromone. It's also involved in the germination in the plant and aging fruit and fruit ripening and environmental tolerance factors. But however, we don't know what is happening in the case of algae. So in order to know that what is this ethylene which does in the algae, so in that way we have targeted similar pathway in the algae. So we have from the methionine, it will be converted into one amino cyclopropane, one carboxylic acid. That will be converted into ethylene. This ethylene plays a major role in the case of stress response. Like during the stress condition, this ethylene will be produced higher condition. So it causes effect in the plant. But in the case of algae, we don't know what is happening. Similarly, so we need to study that what is the ethylene doing. So in order to weigh that in plant, how to overcome this, they are expressing this ACC deaminase in the algae to convert this because of the ACC molecule into ammonia. So in order to know that what is happening in the algae, so what we have done is like from the Pseudomonas foetida, we have took this ACC deaminase gene and we have cloned into PCLAMI4 vector system, like it is an Chlamydomonas expression vector through electroporation with the conditions of voltage of 500 volt and with the capacitance of 50 and resistance of 800 ohm. So then we have transformed after electroporated cells will be transformed into selective plate medium with the zeosin as a selection marker. So that we have used as a 10 mg per liter in the tap plate medium. So then we have incubated and we found that colonies after 48 days. After that, we have done the molecular characterization and enzyme activity, and we have studied the phenotypic characters. This is the DNA we have isolated, and we have found that transgene presence in the ACC deaminase in the transgenic lines. So totally 36 transgenic lines we have randomly we have chosen, selected. So among that, almost 20 to 25 were positive. So from with that, we have found that what is the ACC deaminase activity. We found that three major strains were we have taken for further studies, like particularly that transgenic lines 13, 26, and 28 were used for further studies because it shows that highest ACC deaminase activity. So this is how the construct looks, which has an HSP70 as a promoter and RBCS2 as a terminator here, and BLE is a gene which is responsible for zeosin activity, and ACC deaminase is our target gene. So initially we have checked with the genomic DNA whether the presence of our transgene with the ACC deaminase presence or not. The CBLP is an act as an endogenous gene. Similarly, we have seen that whether the presence of RNA level also, we have found that is R present. Then we have Southern blot analysis to integration of the copy number. So we found that single copy number we have observed in the case of all the transgenic lines. Similarly, as I said in the topic itself, whether it is growing in the nitrogen sorbate condition as well as nitrogen replete condition, and we observed that there is no significant difference we observed in the case of a nitrogen replete condition, whereas in the case of nitrogen deplete condition, we observed that significant decrease in the growth of wild type when compared to the transgenic lines. So even the case of nitrogen replete condition as well as nitrogen starvation condition, how this ACG deaminase activity was increasing. So this shows that that ACC deaminase was producing at the higher condition at the nitrogen deplete condition. It's because of higher amount of ethylene production. So for that conversion rate, it is producing higher. Similarly, we have seen that ethylene production also we have observed that as we seen that enzyme activity, but we didn't see the ethylene production, so that we have took in the culture in the serum vial. So H-phase gas will be evaluated for ethylene production. So the H-phase gas were evaluated the ethylene level in the case of wild type as well as in the case of transgenic lines. We have found that wild type having a higher ethylene content when compared to the transgenic lines. It's because of this ACC deaminase, which is converting this ethylene into ammonia. So that's why the ethylene content was decreased. In the case of both, in the case of nitrogen depleted condition as well as nitrogen depleted condition. Even as we've seen that growth pattern also, which is showing that predominantly we observe that nitrogen solvent condition is can able to grow higher in case of and compared to transgenic lines. So as we've seen, our target is a lipid content. So we have used body pi 505 and 515 as a fluorescent dye we have used for observing the lipid globules in the algae. So in the case of wild type, which shows that lesser in the amount of nitrogen deplete condition and deplete condition also. Whereas in compared to the transgenic line, which shows that higher amount of lipid content and compared to the wild type. So in the case of fluorescence activity through presence of one, we have observed that fluorescent activity also, and the total lipid content we have observed that in the case of wild type as well as in the transgenic lines. So total fatty acid also enhanced in the transgenic line, and the total lipid content also enhanced in the transgenic lines. So these are the some of the whether it is that nitrogen replete condition and deplete condition whether affect the cells or not. So in order to see that, we have seen that ROS and lipid peroxidation level. So ROS we have used DCFCDA so as a marker, and we observed that there was in the nitrogen deplete condition there was a decrease in the ROS level when compared to the control. Similarly, we have observed that the same thing observed in the case of lipid peroxidation level also, it shows that ACC deaminase, which shows that which has an antioxidant activity also, which plays a major role. So similarly, we have observed that antioxidant enzyme activities also SOD, CAT, and APS, APX enzyme proteins also, which have seen that. So in that ACC deaminase activity in the transgenic lines with all the antioxidant enzyme was showed which is enhanced activity in the case of all the antioxidant enzymes. This shows that in the overall study, which shows that compared to the wild type and the ACC deaminase overexpressing line shows that. So it has enhanced the stress tolerance, biomass, overall photosynthetic activity, and lipid production, ROS reduction, and antioxidant defense mechanisms. And thank you. Yeah, have any query?
Thank you, sir, for your informative lecture. Participants, if you have any queries, you can raise them in the chat box. Participants, if you have any queries, you can raise them in the chat box. While we wait for queries from the participants, please note that the Satyabama Center for Ocean Research is organizing its two-year MSc program in marine biology. The admissions are open and the last date for application is 14th of August 2026. The classes for the MSc program in marine biology begins on the 17th of August 2026. The eligibility is a UG degree in life sciences with a minimum of 55% aggregate. The request details for sample analysis and consultancy charge sheets are posted in the chat box. Students, participants availing characterization services for your final year projects or your dissertation works can check the chat box. For further information on programs organized by Center for Ocean Research, trainings and workshops, you can check out our official website and our social media pages on LinkedIn, Facebook, and Instagram. So we do have a set of programs that are coming up. The first program is the National Conference on Artificial Intelligence for Ocean Sciences, organized by Center for Ocean Research in association with INCOIS Hyderabad. The application deadline is 15th August 2026. It's a national conference sponsored by Ministry of Earth Sciences Deep Ocean Mission Project. And coming up next is the 2-day hands-on program, a training program on microplastics in the marine environment, organized by COR. The training dates are 28 and 29 August. And the next program that's coming up is the hands-on training program on zebrafish toxicological experimentation techniques. So that program is being organized from the 18th and 20th of August. Registration links and brochures are being shared in the chat box. Interested participants can register yourselves and participate. As we see, there are no queries in the chat box. We'll close the lecture series for today here. I request all the participants to join back at 2 o'clock for another insightful session. Thank you.